If you take a piece of raw chicken out of your freezer, you probably just assume the freezing temperatures have killed off any dangerous bacteria.
Right. Yeah, I mean, most of us do.
Right, because we think of extreme cold or like extreme heat as this solid physical wall that microscopic life simply cannot cross. But you aren't actually killing those microbes in your freezer. You are just hitting the pause button.
Exactly. And it's a very common and honestly sometimes a very dangerous misconception. We are constantly surrounded by this completely invisible microbial biosphere, and that biosphere is totally beholden to a set of rules. Fences, basically, made of pure chemistry and physics.
Yeah.
But those fences don't work the way our human intuition tells us they do.
And that is exactly our mission for this deep dive. We are exploring the two most critical parameters that dictate those invisible rules, which are temperature and pH.
Right.
We're going to break down how these two factors control the entire microbial universe, how certain microbes manipulate their environments to survive conditions that should literally melt them.
Literally melt them, yeah.
Yeah. And how our own human-centric bias delayed our understanding of all of this for decades.
Which is such a fascinating part of the history. But we also need to look at the practical stakes here, because understanding these environmental fences isn't just, an academic exercise.
Right. It has real-world consequences.
Huge consequences.
Yeah.
Misjudging them has led to massive shifts in medical science, huge technological leaps, and unfortunately some fairly tragic events in the history of the food industry too.
Wow. Okay, so to start, I want to understand what these boundaries actually look like for a single-celled organism, because since they are single-celled, it's not like they can wear coats or sweat.
Right. They don't have those options.
So if the environment gets too hot or too cold, like what actually happens to them physically?
Well, every single species of microbe has what microbiologists call a cardinal range for both temperature and pH. It's basically a biological absolute, and it's defined by three numbers. You have a minimum, an optimum, and a maximum.
Okay, minimum, optimum, maximum.
Exactly. Drop below the minimum or, push past the maximum, and growth just stops. Based on these three cardinal points, we categorize microbes into different groups.
I'm assuming one of those groups includes the microbes that live in and on us, since we are their environment.
Yes, those are the mesophiles. They thrive in moderate temperatures, usually between like 20 and 45 degrees Celsius. But then you have psychrophiles, which actually actively require the cold to survive.
Okay.
And on the other end of the thermometer, you have thermophiles, the heat lovers, and hyperthermophiles, which demand this extreme near-boiling heat.
And I'm guessing there's a pH equivalent for this, too.
Yeah, there's a parallel system for pH, measuring acidity. Acidophiles thrive in heavy acid, neurophiles prefer neutral environments like us, and alkaliphiles demand highly basic conditions.
That sounds like a very tidy, almost symmetrical system on paper. But there is an underlying biochemical engine driving all of this, right? Specifically when it comes to temperature. It has to do with how fast they grow within that cardinal range.
Yeah, it really all comes down to molecular physics. Chemical reaction rates roughly double for every 10 degrees Celsius rise in temperature.
Oh, wow.
Yeah, in biology, this is actually known as the Q10 coefficient.
Okay, let's break that down for the listener. Why does a 10-degree rise double the speed of a biological reaction? That seems like a massive jump.
It is. And you just have to think about kinetic energy. Temperature is essentially just a measurement of how fast molecules are vibrating and moving around, right?
Right.
So when a microbe's environment gets warmer, the molecules inside its cell move faster. They bump into each other way more often. The enzymes, which are, the little protein machines doing all the actual work in the cell, they collide with their targets much more frequently.
So it's just more collisions, more reactions.
Exactly. So as the temperature rises toward that microbe's optimum point, its metabolic engine basically revs faster and faster.
Okay, I picture it kind of like driving a car up a mountain. The climb from the minimum temperature up to the optimum is this steady, ramping incline. The warmer it gets, the faster the car goes.
Yeah, that's a good way to look at it.
But what happens when you hit the peak? Like that optimum temperature and you keep driving, it's not a gentle decline down the other side of the mountain, is it?
No, not at all. It is a catastrophic, completely vertical drop off a cliff.
Wow.
The moment a microbe is pushed just a few degrees past its optimum temperature, growth doesn't just slow down. It plummets to zero, and the cell usually just dies.
But why is the drop-off so sudden? Like, if warmer means faster, why does a little too much warmth instantly kill the cell?
Because the machinery literally falls apart.
Oh.
Yeah, this is called protein denaturation. Enzymes and proteins are these long strings of amino acids folded into highly specific three-dimensional shapes. And those shapes are held together by very delicate molecular bonds, primarily hydrogen bonds.
And in biochemistry, shape is everything, right? Shape is function.
Exactly. If an enzyme loses its shape, it becomes completely useless.
And the heat just like vibrates those delicate bonds apart.
Essentially, yes. The kinetic energy becomes too chaotic, the proteins unravel, but, and this is crazy, it actually gets worse.
Wait, how does it get worse than your proteins unraveling?
Well, a cell is surrounded by a membrane, right? Which is this beautifully organized semi-fluid lipid bilayer. When it gets too hot, that membrane melts. It just turns into a leaky puddle.
Oh, wow. Which means the cell can't keep the outside world out or the inside world in.
Exactly. And it loses its ability to generate energy because a cell is essentially just a tiny battery. It pumps charged ions across its intact membrane to store energy.
So if the membrane melts into a puddle,
The battery shorts out. It is lethal. Take E. coli, for example. Its minimum gross temperature is around 8 degrees Celsius. The peak of its mountain, its optimum, is human body temperature, 37 degrees.
okay, that makes sense.
But its absolute maximum is just 45 to 49 degrees.
Wow.
Yeah, it takes nearly 30 degrees to climb the mountain, but only about 10 degrees to fall off the cliff and die.
That is such a tight window. But, if these boundaries are so strict, how did early scientists map them out? Because if we are sitting here at 37 degrees Celsius, I imagine early doctors just assumed all important life operated at our temperature.
Oh, they totally did. 19th century medical bacteriology was built almost entirely around studying pathogens that make humans sick. So there was this massive human-centric bias.
Right.
Normal biology was defined simply as what was normal for us.
So how did we finally break out of that bias?
Well, it started with some really surprising observations in the late 1800s.
Yeah.
In 1887, a bacteriologist named Wilhelm Forster isolated bacteria from North Sea fish, and he found that they were actively multiplying at 0 degrees Celsius,
Case in point.
literally at the freezing point.
That completely shatters the assumption that bacterial life requires warm-blooded heat.
It did. It forced a total paradigm shift. And then around the same time, Pierre Michel began pulling heat-loving bacteria out of the Seine River in France.
No way.
Yeah. He found microbes thriving at temperatures that would boil E. coli alive. It really pushed the scientific community to realize that normal is entirely relative to the specific organism.
And it wasn't just temperature where we had these blind spots, right? The history of how we started measuring pH is so fascinating. The pH scale is basically foundational to modern science. You see it in every medical show and chemistry textbook to measure how acidic or basic something is.
Right.
Did a doctor invent that to measure human blood acidity?
Not at all. It was actually invented in 1909 at the Carlsberg Brewing Laboratory in Copenhagen.
Wait, really? The pH scale was invented to make better beer.
It was. It was a biochemist named Søren Sørensen. He was trying to understand the behavior of brewing yeast and the enzymes involved in fermentation,
Okay.
because the older, cruder ways of measuring acidity just weren't precise enough for the brewery. The yeast was way too sensitive to slight changes in the environment.
So just to guarantee a consistent pint of lager, he creates a brand new system of measurement. That's amazing.
Yeah, he introduced the concept of pH potentia hydrogenii, the power of hydrogen. And it gave scientists a standardized logarithmic scale to correlate exactly what the microbes were doing with the precise concentration of hydrogen ions in their environment.
That is a brilliant historical pivot. And as we got better at measuring these things, we started pushing the boundaries even further. The timeline in our sources points to the 1960s as the dawn of the extremophile era.
Yeah, that was when we moved from just observing the occasional oddity to actively hunting for the outer limits of life. The pivotal figure here is Thomas Brock.
Right, in 1966.
Yeah, 1966. He was out in Yellowstone National Park taking samples from the bubbling hot springs.
Water that is practically boiling. Scientists universally assume those pools were totally sterile, right?
Absolutely. The assumption was that the biochemical cliff we talked about earlier made life in those springs physically impossible. But Brock found bacteria thriving in them. By 1969, he and his colleague Hudson Freeze had described an organism they called Thermus aquaticus. And this organism grows optimally at around 70 degrees Celsius.
Which is far past the melting point for most cellular machinery. And this wasn't just a neat piece of biological trivia either.
No, it completely revolutionized modern genetics. Because Thermus aquaticus evolved in near boiling water, its internal machinery doesn't denature at high heat.
Oh, right.
And that includes its DNA copying enzymes. So in the 1980s, scientists isolated a heat-stable enzyme from it called Taq polymerase. They used that enzyme as the engine to invent PCR, the polymerase chain reaction.
Which is the technology used in crime scene forensics, genetic testing, and literally every COVID-19 swab. All of it traces back to a microbiologist getting curious about a bubbling hot spring.
It's wild, isn't it?
It's incredible. But it does bring up a massive mechanical question. If human proteins melt at 45 degrees, how is that Yellowstone bacteria surviving at 70 degrees? Like, what is its cellular survival toolkit?
Well, the adaptations are incredibly elegant. One of the most fascinating mechanisms is called homeoviscous adaptation, which is a concept outlined by Mark Sinensky in 1974. And it essentially solves the problem of that lipid cell membrane turning into a leaky puddle.
Right, how do they prevent the membrane from melting or like from freezing solid in the cold?
They actually act like microscopic chemists tuning a lubricant. To understand this, think about the difference between butter and olive oil in your kitchen.
Okay, butter is solid at room temperature and olive oil is liquid.
Because of their chemical structure, butter is made of saturated fatty acids. The molecules are perfectly straight, so they pack tightly together, which keeps it solid.
Right.
Olive oil is made of unsaturated fatty acids. The molecules have these little kinks in them, which prevents them from packing tightly so it stays fluid even when it's cold.
Wait, so a microbe can change its membrane by swapping these out?
Yes, in real time.
Wow.
If the environment gets dangerously hot,
Mm-hmm.
the microbe packs its membrane with straight saturated fatty acids like butter to keep the membrane from melting.
Oh, I see.
And if the environment drops toward freezing, it swaps them out for kinked unsaturated fatty acids like olive oil so the membrane stays perfectly flexible. They actively re-engineer their physical boundaries to match the temperature.
That is genius. Okay, so that explains temperature. But what about pH? Let's say you have an acidophile living in a volcanic soil that is as corrosive as battery acid. Do they have acid-proof machinery inside their cells?
Surprisingly, no. Their internal enzymes are just as sensitive to acid as ours are.
Really?
Yeah. Surviving extreme pH is basically a trick of the cell envelope. Acidophiles are not acidic on the inside. Their cytoplasm remains near neutral. They maintain this really strict internal homeostasis.
So the outer envelope acts like a biochemical hazmat suit.
Basically.
But how does it actually keep the acid out? Because acid is just a high concentration of hydrogen ions, right? Don't those ions naturally just leak into the cell?
They do leak in. The hazmat suit isn't just a passive shield. The cell is essentially bailing water out of a sinking boat. It uses these specialized transport pumps to actively shove those acidic protons back out into the environment as fast as they leak in. All the extreme tolerance is in the outer envelope and those specific pumps.
That sounds absolutely exhausting for the cell.
It requires a massive amount of energy. But perhaps the most wild example of manipulating pH doesn't involve keeping acid out, but neutralizing it entirely.
Okay.
This is the story of Helicobacter pylori.
Ah, yes. Human stomach acid has a pH of, what, 1.5 to 2? It is brutally corrosive, designed to break down a steak and kill pathogens. And I know for decades, medical dogma said the stomach was totally sterile.
The consensus was that literally nothing could survive that acid bath. So when patients developed stomach ulcers, doctors just blamed stress or spicy food.
Right.
But in the early 1980s, two researchers, Barry Marshall and Robin Warren, identified a bacterium, H. pylori, living right in the lining of the stomach.
And the medical establishment basically laughed them out of the room, right? They completely rejected the idea.
The dogma was just too deeply entrenched. But so in 1984, Barry Marshall decided the only way to prove his theory was a dramatic self-experiment.
Oh, boy.
He deliberately drank a murky broth teeming with cultured H. pylori.
Wait, he chugged the bacteria just to prove they could survive his own stomach acid?
He did, and he developed severe, extremely painful gastritis.
Unbelievable.
The bacteria successfully colonized his stomach. He proved definitively that a microbe, not stress, was the cause. He later cured himself with antibiotics, and he and Warren actually won the 2005 Nobel Prize for this discovery.
It's an incredible story of scientific rebellion, but what is the specific trick here? Like, how did the bacteria he drank not dissolve instantly in his stomach?
H. pylori produces massive amounts of an enzyme called urease. Urease takes urea, which is naturally present in the stomach anyway, and splits it into carbon dioxide and ammonia.
And ammonia is a highly basic alkaline compound.
Is it right?
So the bacterium wraps itself in a neutralizing cloud of ammonia.
Yeah, it creates this tiny, safe, perfectly neutral microenvironment right in the middle of a vat of boiling stomach acid.
It literally builds its own invisible fence.
It does.
Now, Barry Marshall proved that underestimating microbial survival can delay life-saving medical treatments. But when we look at the food industry, misjudging these temperature and pH limits has even higher stakes.
It can be lethal. The 1971 Bon Vivant Vichyssoise poisoning is kind of the tragic case study here.
Like, what happened there?
So a batch of canned potato and leek soup was improperly thermally processed at the factory.
Meaning they didn't cook it hot enough?
Exactly. It didn't reach the required temperature for the necessary duration.
And because of that failure, spores of Clostridium botulinum survived inside the sealed cans.
Right. And a sealed can has basically no oxygen, which is the exact environment botulinum needs to thrive. The spores germinated, multiplied, and produced botulinum toxin.
That's terrifying.
It resulted in at least one death, severe illnesses, and the complete collapse of the Bon Vivant company.
How does the modern canning industry guarantee that never happens again? Because if bacteria can build ammonia clouds and survive boiling springs, how do we ever guarantee a can of soup is actually safe?
Well, they rely on a unified safety threshold called the botulinum cook, which treats temperature and pH as interacting variables. It all comes down to acidity.
Okay, how so?
If you are canning a low-acid food like that potato soup or green beans, basically anything with a pH above roughly 4.6, you
Yeah.
have to use extreme heat. Specifically, a thermal process equivalent to 121 degrees Celsius for a set duration.
Wow, 121.
Yeah, that is the global standard required to physically destroy botulinum spores.
So because the food isn't acidic enough to stop the bacteria, you have to nuke it with heat. But what if you are canning a high-acid food like, say, pickles?
If the food falls below that 4.6 pH threshold, you don't need that extreme 121-degree heat treatment. The acidic environment itself physically prevents the botulinum spores from germinating and producing the deadly toxin, even if the spores are technically present. Yeah, the pH does the heavy lifting, so the temperature doesn't have to.
That makes a lot of sense. But I need to pause on that 121 degrees Celsius number.
Okay.
Because if 121 degrees is the global medical standard, like the temperature that every autoclave and every laboratory uses to sterilize equipment, because it's the literal temperature that kills everything, what does it mean that researchers recently found something called strain 121?
Yeah, you're referring to Kashafi and Lovely's discovery in 2003. It was a massive shock to microbiology. They pulled an iron-reducing microbe from a deep-sea hydrothermal vent, and to test its limits, they put it in an autoclave at exactly 121 degrees Celsius.
The ultimate death chamber for microbes.
Right. They left it in there for 10 hours.
Oh my God.
And not only did the microbe survive the sterilization process, it reproduced. It actually doubled its population.
Inside the sterilization oven.
Inside the oven, it forced science to reconsider a fundamental assumption. Like, is the upper limit of life a real, fixed biochemical ceiling? Or is it simply a reflection of the extreme environments we just haven't bothered to sample yet?
If 121 degrees isn't the absolute ceiling we thought it was, it really makes me wonder what else we are getting wrong in our day-to-day lives. Our sources highlight several pretty dangerous misconceptions people have about temperature and food safety in their own kitchens.
Oh, definitely. The fridge myth is perhaps the most common one. People put leftovers in the refrigerator, which sits at about 4 degrees Celsius, and they assume the cold kills the bacteria, rendering the food safe indefinitely.
But based on what we established earlier, it just drops the temperature below the optimum for those mesophilic bacteria.
Exactly. It moves them down the slope of that Arrhenius curve. It slows their metabolic engine down. It does not kill them. And worse, some dangerous pathogens are psychrophiles, meaning they tolerate the cold remarkably well.
Like what?
Listeria monocytogenes is the classic example. It will happily continue to multiply at refrigerator temperatures. That is exactly why deli meats have strict use-by dates, regardless of refrigeration.
What about the freezer? If the fridge just slows them down, surely tossing raw meat into a deep freeze at negative 20 degrees destroys the cells, right?
No, freezing halts growth completely, but not by destroying the cell. It halts growth by locking up all the water as ice, making it biologically unavailable.
Okay.
The engine is just paused. The moment you take that raw chicken out and let it thaw on the counter, the water becomes liquid. The temperature rises back into the permissive zone, and those bacteria wake right back up and resume multiplying.
So freezing is hibernation. It is not sterilization. It is
Exactly.
a very crucial distinction to keep in mind. Now, there is another misconception here called the optimum myth. Logically, if a bacteria is growing at its optimum temperature, reproducing the fastest, that must be when it is most dangerous,
That
right?
seems intuitive, yeah. But optimum growth rate and maximum toxin production are not always the same thing. Some pathogens actually switch on their toxin-producing genes, or they accumulate more dangerous byproducts at temperatures slightly below their fastest growth optimum.
Really?
Yeah, food safety guidance actually has to account for these complex metabolic shifts, not just simple growth rate curves.
Taking all of this in, from the deep freeze to strain 121 multiplying in an autoclave, where does the boundary actually end? Like, is there an ultimate ceiling for biology?
That is currently a major debate in astrobiology. I mean, some biochemists argue there must be a hard ceiling, theoretically around 150 degrees Celsius,
The ultimate biochemical cliff.
right, where DNA and proteins just inherently fall apart no matter how many saturated fatty acids or heat-stable adaptations a cell possesses.
But let me guess, other scientists disagree.
Totally. Others point to the history we just discussed. We keep revising the limit upward every single time we explore a new extreme environment on Earth. And this debate directly dictates where astrobiologists point their instruments.
How so?
Well, if you're looking for extraterrestrial life in the subsurface oceans of icy moons like Jupiter's Europa or Saturn's Enceladus, you use our earthly extremophiles to define the habitable envelope.
We've covered incredible ground today. Temperature and pH truly are the invisible puppet masters of the microbial world. They dictate everything from how we brew beer and amplify DNA to how we treat stomach ulcers and ensure our canned food doesn't carry botulism.
They really are the foundational parameters of biology.
Yeah.
You simply cannot understand microbial life without understanding where its limits lie and the elaborate biochemical tools it uses to fight for survival within them.
But I want to leave you with a final thought, something kind of hidden in the mechanisms we discussed today that shifts the entire perspective.
Yeah.
We tend to think of extremophiles as simply enduring these hostile environments,
Right.
like enduring the boiling heat or enduring the battery acid. But organisms like acidophiles don't just put on a biochemical hazmat suit to tolerate the acid, they actually use it. They exploit the massive difference between the highly acidic environment outside their cell and the calm, neutral environment inside their cell. They let the acidic protons rush in through specific channels, and they use that rushing current to physically spin a microscopic turbine in their membrane to generate their own cellular electricity.
It's an incredible mechanism, chemiosmosis. The extreme environment isn't an obstacle to overcome. It is the power source itself.
So next time you're putting away groceries, trying to keep your food out of that temperature danger zone, ask yourself, are microbes just fragile organisms that need to be protected from extremes? Or are they microscopic engines waiting for exactly the right extreme conditions to truly thrive?